name is Lindy Humphrey, and this is my journal club for 3110, which is electromyography of the human nasal muscles. In this video, these are the different things I will be going over of this study to be able to describe each one. First, the description. This study examines the use of EMG to selectively record the activity of human nasal muscles. Specifically, the muscles observed include the dilator nares, the nasalis muscle, or the alar and transverse parts, and apius nasi. The purpose was to see what muscles in the nose strongly relate to respiratory function, and more significantly, what muscles contribute to the prevention of collapse in the nasal valve. Some of the research questions in this study include, first, how to establish the suitability of the investigative method, as before the study, they weren't sure if they would be able to measure this in the different nasal activities. From there, what is the role of the nasal muscles in respiration, and is there a relationship between muscle activity and nasal patency? Lastly, the researchers inferred that a longer, narrower nose would be more prone to alar collapse, and they wanted to see if this is true, if the form influences the presence of muscle activity. In this study, EMG was used to measure the activity in different regions of the nose using bipolar skin surface electrodes. These were placed on the nasal skin to be able to read the movements of certain muscles, as stated before, to understand the movement of these different muscles during different activities, and thus understand a connection between these and the function of the nose. Next, the literature evaluation by assessing the literature references. Reading through the references, and at the bottom of the study, I can tell that the conductors did a lot that they went through and had good references that connected everything. They had many references connecting them to the physiology of the nasal muscles, showing a lot of background research for their understanding and to properly reference information. And they also had references about the nasal pathway during respiration or different activities, the activation and reflex responses in the upper respiratory tract. These references directly relate to the research questions, as well as they show a lot of background information and that the researchers can back up their readings. Next, the subjects. These included 17 healthy adult Caucasian males, and they were aged between 19 and 34 years old. The study also identified that none of the participants had nasal complaints or a history of nasal surgery, showing some exclusion criteria of the study. As it is suggested, the researchers asked the participants if there were prior issues in their nose that would keep them from the study. The inclusion reasoning, or the reasoning that these specific user groups weren't very clear, and it does raise some selection bias concern. Because of looking at these participants, the user groups are not very representative, as other groups may have shifted the results. So there is evidence of bias in the study, as well as in the fact that this is an older study, showing more reason for why that there could be bias. Next, method and design. First, electrodes were placed on the patient's nasal skin, which had been cleaned with alcohol and ether. They created 6 electrode pairs to record muscle activity in the 6 different nasal muscles that were being recorded. And then also shown in the figure I added at the top again, showing the placement. They are placed along the length of the muscle, and then they also gave electrode gel to lower and stabilize surface skin resistance. A ground electrode was placed in the neck, and then EMG signals were pre-amplified using differential amplifiers and continuously monitored. They had patients sit in comfortable chairs and had 7 different conditions that they recorded measurements in to show the different connections. The recording time for each was 30 seconds and then a rest period of 30 seconds. At the end, the subjects were asked to grimace or scrunch their nose to achieve maximum voluntary action of the nasal muscle. Then for data collection, the presence or absence of the EMG activity in the particular muscle was scored. Specifically, statistical analysis, crosstabs were used with the Fisher exact test. The chart readings of all movements were compared with the reading of maximum activity. Taking into account both duration and amplitude of the signals, EMG activity for each movement was normalized based on maximum activity. These results were averaged over all subjects. All adjacent recording sites were compared in each of the 15 subjects. And then overall, all of the data was placed into charts and tables to be analyzed using cross-correlation analysis as well. And then here's a better picture of that figure to show the placements on the different muscles, also showing how the study can be replicated. And then analysis. The study uses figures and tables to describe the different EMG readings and compare the results. Table 1 lists the mean value of the cross-correlation coefficients between EMG signals of the different muscles. Only those EMG signals recorded from adjacent muscles were compared. The table shows that during simultaneous contraction of adjacent muscles, the electrode pairs on these muscles had low cross-correlation. Table 2 summarizes the results for each subject's quiet nasal breathing, quiet oral breathing, and nasal breathing after exercise. For each individual, the presence of inspiratory EMG activity is listed. Muscle activity was found in this phase in 8 out of the 17 subjects during nasal and oral breathing at rest. The location of the muscle activity differed to some extent, but usually the active muscles were pretty consistent. And then, in each subject, the number of active muscles were increased, but all-hour movement was never seen. Table 3 showed that the nasal muscle activity was more present in the group with a high nasal resistance. The observation was statistically significant using Fisher's exact test and a p-value of less than 0.05. There is no relationship between the muscle activity and leporine, which is just the narrowness of the nose. And then these figures also break down these readings. Figure 3 shows all raw EMG signals during nasal flaring, and the level of activity varied from one muscle to another. And then the means of the relative EMG activity were computed over all the subjects, and those results were depicted in Figure 4. As seen in Figure 4, voluntary nasal movements were accompanied by a pattern of activity in all 6 muscles. And then during breathing, those 2 muscles specified were showing sporadic EMG activity. Overall, these tables and charts show both the data in a concise way that the researchers were able to analyze and get significant data from. These tests were also significant as they showed a better comparative state between the different nasal muscles during different activities, showing how they work and how much they work during different activities. And since they show selective recording of the nasal muscles is possible, the study results are significant. And then we go into the findings. The findings of the study demonstrated that the selective recording of the nasal muscle activity is possible, as well as showing which muscles play a role in respiration, with the presence of muscle activity. Their respiratory function is to prevent all-hour collapse during inspiration. Their study showed using multi-point measuring technique that apart from the dilator nares, both alar and transverse parts of the nasal muscle and the APS nasi are active in the inspiratory phase of respiration, unlike that from other EMG studies. The description of the tables and figures, as well as the discussion at the end, connect to explain the data and was presented clearly and understandably, so I could also describe them in this presentation as well. The authors interpreted the results by comparing the tables they created with previous data to show the difference in the findings and what they had and why that was important. The researchers do point out that there has been some speculation concerning the function of the dilator nares, and they explain that this upper airway muscle of respiration acts by adduction of the vocal cords during inspiration, keeping the upper airway open. Since they know that other nasal muscles also may be active during inspiration, it is likely that the various nasal muscles also work together in influencing rigidity of the nasal ala, and then maintenance of the upper airway patency. And then another big limitation of the study could be that the study group bias in the year in which this study was conducted being a bit older. Finally, the significance. Some implications of the study include understanding how different nasal muscles work and how researchers can interact with different nasal muscles if there is a respiratory problem. Being able to selectively measure nasal muscles can, in the long run, give very helpful insight to researchers. This study contributes to the way researchers are able to understand how nasal muscles work and the different roles that they can play. This study definitely can be replicated from the different charts and figures, as well as how they are interpreted and why. And there were some additional questions from the study. Thank you so much for watching, and I also made sure to cite the study here on this slide.